Good day, medical scientists of the future. This is week 2 in Clinical Bacteriology lecture series. My name is Julie J. Liu, and I'll be your lecturer for the topics on bacterial growth and genetics. Just a few reminders before continuing this lecture. First, make sure you are situated in a space where there are no distractions. You are in a learning mode state. Second, make sure you have eaten well and have emptied your bladder before continuing this lesson. Third, you have downloaded the PDF copy of this presentation via Quipper School. You must have a pen with you now to write down additional notes to better understand the bulleted phrases in the handout. And fourth, you are aware that you need to finish studying these lessons before September 9th, because the summative quiz for week 2 lessons will be conducted on Wednesday, September 9th. Lesson 3, Part 1 is on physiological and nutritional growth requirements of bacteria. At the end of this lecture, the student is able to identify the growth requirements of bacteria and discuss the importance of the physiological and nutritional requirements for bacterial growth. How do we grow bacteria? And why do we have to know how to cultivate or grow them? What equipment and laboratory materials do we need? So look at this first image. You have your incubator, and do all bacteria grow at this temperature at 37 degrees Celsius? Why do we need some of them to be placed in a gas pack system, like in the second picture at the center? And on the third, look at this jar, and inside it there's a candle. If we lit this candle, this will emit carbon dioxide inside a bottle. So there are certain bacteria that may require carbon dioxide. Why do we need to grow them in a solid medium, an agar-based medium? No? And what are the ingredients needed for them to grow? To grow bacteria, we need to study their physiology. Every organism must find in its environment all of the substances required for energy generation and cellular biosynthesis. The chemicals and elements of this environment that are utilized for bacterial growth are referred to as nutrients or nutritional requirements. In the laboratory, bacteria are grown in culture media, which are designed to provide all the essential nutrients in solution for bacterial growth. The nutritional requirements of a bacterium are revealed by the cell's elemental composition, which consists of major and trace elements. And these elements are found in the form of water, inorganic ions, small molecules, and macromolecules, which serve either a structural or functional role in the cells. Now, the major elements for a bacterium, 50% of its dry weight constitutes carbon. We know carbon is the main constituent of cellular materials. Oxygen is about 20%. As we all know, oxygen is an electron acceptor in aerobic respiration. Nitrogen is about 14% of a bacterium's dry weight. It's a constituent of amino acids, nucleic acids, nucleotides, and coenzymes. Hydrogen, about 8%. It's the main constituent of organic compounds and cell water. Phosphorus, 3%. It's also a constituent of nucleic acids, nucleotides, phospholipids. In the cell wall of bacteria, they are found attached to the lipopolysaccharides and teichoic acids. Sulfur, about 1%, and it's the main cellular inorganic cation and cofactor for certain enzymes. Magnesium is about 0.5%. It's an inorganic cellular cation, acts as a cofactor for certain enzymatic reactions. Calcium has the same physiological function. It's about 0.5%, and it also acts as a cofactor for certain enzymes and a component of endospores. Iron, 0.2% only. It's a component of cytochromes and certain non-heme iron proteins and a cofactor for some enzymatic reactions. On the other hand, the trace elements are metal ions required by certain cells in just small amounts. These amounts are very difficult to measure. It's not necessary to add them to culture media as nutrients. Trace elements are required only in such small amounts that they are present as contaminants of the water or other media components. So we have trace elements manganese, cobalt, zinc, copper, and molybdenum. All living organisms require a source of energy. Organisms that use light or radiant energy are called phototrophs. These organisms derive energy by photophosphorylation. Organisms that use or oxidize an organic or inorganic form of carbon are called chemotrophs. These organisms derive energy by oxidative phosphorylation. Organisms that obtain their reducing power from inorganic compounds are called chemolithotrophs, while organisms that obtain their electrons from organic compounds are called chemoorganotrophs. Organisms that use carbon dioxide as a sole source of carbon for growth are called autotrophs. On the other hand, organisms that use organic carbon are heterotrophs. Thus, on the basis of carbon and energy sources for growth, four major nutritional types of prokaryotes may be defined. Some of our purple and green bacteria that are non-oxygen producers belong to photoautotrophs. Of course, our oxygen producers like cyanobacteria and some algae and even plants belong to this group as well. Some of our purple and green bacteria also belong to photoheterotrophs. Majority of our archaea and some iron, sulfur, and hydrogen and nitrifying bacteria also belong to chemoautotrophs or lithotrophs, while the majority of our pathogenic bacteria, most bacteria, some archaea, and of course animals, protozoa, fungi, belong to chemoheterotrophs or are heterotrophs. Some chemoheterotrophs may require small amounts of certain organic compounds for growth because they are essential substances that the organism is unable to synthesize from available nutrients. Such compounds are called growth factors. Growth factors are required in small amounts by cells because they fulfill specific roles in biosynthesis. The need for a growth factor results from either a blocked or missing metabolic pathway in the cells. Growth factors are organized in three categories: purines and pyrimidines. These are required for synthesis of nucleic acids like DNA and RNA. Amino acids. These are required for synthesis of proteins. Vitamins. These are needed as coenzymes and functional groups of certain enzymes. Some bacteria, like E. coli, do not require any growth factors. They can synthesize all essential purines, pyrimidines, amino acids, and vitamins, starting with their carbon source as part of their own intermediary metabolism. But certain other bacteria, like the lactobacillus, they require purines, pyrimidines, vitamins, and several amino acids in order to grow. These compounds must be added in advance to culture media that are used to grow these bacteria. The growth factors are not metabolized directly as sources of carbon or energy. Rather, they are assimilated by cells to fulfill their specific role in metabolism. And in bacteriology, we call such organisms as fastidious. Fastidious bacteria, such as Haemophilus influenzae, tend to require a variety of growth factors. How about physical and environmental requirements for microbial growth? The prokaryotes exist in nature under an enormous range of physical conditions such as oxygen concentration, hydrogen ion concentration or pH, and temperature. Let's start with the effect of oxygen. Oxygen is a universal component of cells and is always provided in large amounts by water. However, prokaryotes display a wide range of responses to molecular oxygen. Bacteria that require oxygen for growth are called obligate aerobes. They use oxygen as a final electron acceptor in aerobic respiration. Those that do not need or use oxygen as a nutrient are called obligate anaerobes. In fact, oxygen is toxic to them. Oxygen either kills or inhibits their growth. Obligate anaerobic bacteria may live by fermentation, anaerobic respiration, bacterial photosynthesis, or the novel process of methanogenesis. What about the term facultative? Facultative anaerobes can switch from aerobic to anaerobic type of metabolism, or facultative aerobes can switch from anaerobic to aerobic type of metabolism. Under anaerobic conditions, no oxygen, they grow by fermentation or anaerobic respiration, but in the presence of oxygen, they switch to aerobic respiration. Facultative. What about aerotolerant anaerobes? Now, these are bacteria with an exclusively anaerobic or fermentative type of metabolism, but they are insensitive to the presence of oxygen. They live by fermentation alone, whether or not oxygen is present in their environment. Okay? And what about microaerophilic organisms? Now, these require oxygen to survive, but only at a lower concentration, about 20% concentration of oxygen. Low levels of oxygen concentration are needed to survive. These are called microaerophiles. Many microaerophiles are also capnophiles. What are capnophiles? They require an elevated concentration of carbon dioxide. Now, in the laboratory, they can be easily cultivated in a candle jar. a container into which a lit candle is introduced before sealing the airtight lid. The flame burns until extinguished by oxygen deprivation, creating a carbon dioxide-rich, oxygen-poor atmosphere. But if you will be working somewhere in PGH or at St. Luke's, they already have a so-called carbon dioxide incubator, so you do not need the candle jar. Let's say you've inoculated your unknown organism into a liquid broth, and after 24 hours you'd like to observe the growth. You would notice some turbidity or precipitation or signs of growth inside the test tube or the liquid broth medium. So here, if they are obligate aerobes, turbidity or signs of growth will appear at the surface level, topmost level of the broth medium, while the facultative anaerobes, you see a homogeneous turbidity in the medium, while for obligate anaerobes, you see signs of precipitation or turbidity at the bottom of the medium, while for aerotolerant anaerobes, just the same, no? Homogeneous growth inside the medium. Those that require just low level of oxygen tension. So see here, the microaerophiles are seen just in the subsurface, no? Level of the broth. How come aerobes and aerotolerant anaerobes survive the lethal accumulation of superoxide? And how come obligate anaerobes cannot survive in the presence of these oxygen radicals? Well, they have enzymes that protect them. Nearly all organisms which can live in the presence of oxygen contain superoxide dismutase to form superoxide into oxygen and hydrogen peroxide, while nearly all bacteria contain catalase that can break down hydrogen peroxide into water molecules. On the other hand, aerotolerant bacteria like streptococci have peroxidase that can derive electrons from NADH to reduce peroxide to water. So most obligate anaerobes or facultative anaerobes like the enterics, E. coli, Proteus, Enterobacter, Klebsiella, they all contain superoxide dismutase and catalase, while streptococci or most aerotolerant anaerobes contain superoxide dismutase and peroxidase. And that's why the Clostridia group, the strictly anaerobes, cannot survive oxygen because they don't have any of these enzymes. Bacteria are subdivided into several classes on the basis of their temperature requirement for growth. The cold-loving organisms are called psychrophiles. They are defined by their ability to grow at 0 degrees. A variant of a psychrophile which usually has an optimum temperature of 10 to 15 degrees, is a psychotroph, which grows at 0 degrees but displays an optimum temperature in the mesophilic range, nearer room temperature. Psychotrophs are the scourge of food storage in refrigerators since they are invariably brought in from their mesophilic habitats and continue to grow in the refrigerated environment where they spoil the food, but they grow slower inside the refrigerator. Psychrophilic bacteria are said to be adapted to their cool environment by having largely unsaturated fatty acids in their plasma membranes. On the other hand, organisms with an optimum temperature near 37 degrees, or the body temperature of warm-blooded animals, are called the mesophiles. So most pathogenic organisms belong to this category. Organisms with an optimum temperature between 45 degrees and 70 degrees are thermophiles, and some members of the archaea have an optimum temperature of 80 degrees or higher, or even as high as 115 degrees. And so they are now called extreme thermophiles or hyperthermophiles. But don't forget, most of the organisms that we grow in the laboratory belong to the mesophilic group. Water is the solvent in which the molecules of life are dissolved, and the availability of water is therefore a critical factor that affects the growth of all cells. The availability of water for a cell depends upon its presence in the atmosphere, and that is what we call as relative humidity, or its presence in solution or a substance. That is what we call as water activity. The water activity of pure water is 1.0. That's 100% water. Water activity is affected by the presence of solutes such as salts or sugars that are dissolved in the water. The higher the solute concentration of a substance, the lower is the water activity, and vice versa. Microorganisms live over a range of water activity from 1.0 to 0.7, and the water activity of human blood is 0.99. The only common solute in nature that occurs over a wide concentration range is salt, or sodium chloride, and some microorganisms are named based on their growth response to salt. Microorganisms that require some sodium chloride for growth are called halophiles. Mild halophiles require 1 to 6% salt. Moderate halophiles require 6 to 15% salt. Extreme halophiles that require 15 to 30% sodium chloride for growth are usually found among the archaea group, and bacteria that are able to grow at moderate salt concentrations, even though they grow best in the absence of sodium chloride, are called halotolerant, like Vibrio cholerae, although halophiles are also called osmophiles, and Halotolerant organisms are osmotolerant. The term osmophiles is usually reserved for organisms that are able to live in environments high in sugar. Organisms which live in very dry environments made dry by lack of water are called xerophiles. So the concept of lowering water activity in order to prevent bacterial growth is the basis for preservation of foods by drying in sunlight or by evaporation or by addition of high concentrations of salt or sugar. When bacteria consumes nutrients for growth, this would depend upon the water activity and solute concentration. There is either simple diffusion and facilitative diffusion across membranes from an area of high solute concentration to an area of low concentration. On the other hand, high water concentration inside or outside the bacterial cell can also have an effect in its growth or survival due to osmosis and osmotic pressure. So that if we want to grow bacteria and let it survive longer, they must be placed in an isotonic solution. In a hypotonic solution, extracellular water enters the cell that may cause the cell to burst. On the other hand, in a hypertonic solution, cytoplasmic or cellular water moves out of the cell and causes it to shrink. Thus, bacteria are killed if they are either placed in a hypotonic or a hypertonic solution. There is essence in why we preserve food in high salt or sugar concentration because hypertonic environments cause bacteria to undergo plasmolysis. However, we need to be aware that extreme halophiles, those that can grow in seawater, can actually survive in an environment with an increased osmotic pressure. What about the effect of pH on microbial growth? The pH, or hydrogen ion concentration, of natural environments varies from about 0.5 in the most acidic soils to about 10.5 in the most alkaline lakes. Most free-living prokaryotes can grow over a range of 3 pH units, about a thousand-fold change in hydrogen. The range of pH over which an organism grows is defined by three cardinal points: the minimum pH, below which the organism cannot grow; the maximum pH, above which the organism cannot grow; and the optimum pH, at which the organism grows best. For most bacteria, there is an orderly increase in growth rate between the minimum and the optimum, and a corresponding orderly decrease in growth rate between the optimum and the maximum pH, reflecting the general effect of changing hydrogen ions on the rate of enzymatic reaction. Microorganisms which grow at an optimum pH well below neutrality, that is below pH 7, are called acidophiles, and they grow optimally between pH 2 to 5. Those which grow best at neutral pH are called neutrophiles. So most bacteria belong to this category. Most bacteria's optimum pH for growth is at 7.2 to 7.6, while those that grow best under alkaline conditions are called alkaliphiles, with optimum at pH 8 to 9. In the construction and use of culture media, one must always consider the optimum pH for growth of a desired organism and incorporate buffers in order to maintain the pH of the medium in the changing milieu of bacterial waste products that accumulate during growth. Many pathogenic bacteria exhibit a relatively narrow range of pH over which they will grow, and most diagnostic media for the growth and identification of human pathogens have a pH near 7. So remember that. So we're finished with part 1 of lesson 3 about physical and nutritional requirements for bacterial growth. Hope you can recall the essential substances needed by bacteria to survive, the major and trace elements. Where do they get their carbon and energy sources? How do you call bacteria that will require growth factors? What enzymes do obligate aerobes and aerotolerant bacteria have? And how do you call bacteria that require increased carbon dioxide tension? And how about temperature? At what category? What is the optimum temperature to grow most pathogenic bacteria? What about water activity and solute concentration? How does it affect microbial growth? As well as remember to know the optimum pH for the growth of a particular bacterium, because you need to incorporate buffers for the optimum pH when constructing or when cooking a culture medium. We go to the next topic, which is on bacterial growth curve. At the end of this lecture, I hope each student is able to describe the different phases of the bacterial growth curve. There are 4 of them. Name appropriate culture media to grow a particular group of bacteria, and summarize the principles involved in culturing bacteria. So we've been talking about bacterial growth, but bacterial growth here means not in the context of size, but more in the context of number. But growth may also mean an orderly increase in the quantity of cellular constituents. Most bacteria reproduce by a relatively simple asexual process called binary fission. Each cell increases in size and divides into two cells. During this process, there is an orderly increase in cellular structures and components, replication and segregation of the bacterial DNA, and formation of a septum or cross wall which divides the cell into two progeny cells. The process is coordinated by the bacterial membrane, perhaps by means of the mesosomes. The DNA molecule is believed to be attached to a point on the membrane where it is replicated. The two DNA molecules remain attached at points side by side on the membrane, while new membrane material is synthesized between the 2 points. This draws the DNA molecules in opposite directions, while new cell wall and membrane are laid down as a septum between the two chromosomal compartments. When septum formation is complete, the cell splits into two progeny cells. The time interval required for a bacterial cell to divide, or for a population of bacterial cells to double, is called the generation time. Generation times for bacterial species growing in nature may be as short as 15 minutes or as long as several days, and the generation time will vary when bacteria are grown artificially under standard nutritional conditions or when they grow inside a host. Example: the generation time of E. coli in the laboratory is 15 to 20 minutes, but in the intestinal tract, the coliform's generation time is estimated to be 12 to 24 hours. When growing exponentially by binary fission, the increase in a bacterial population is by geometric progression. If we start with one cell when it divides, there are two cells in the first generation. Remember that, two cells. Then four cells in the second generation, eight cells in the third generation, and so on. The generation time is the time interval required for the cells or population to divide. Let's try this case. If a single bacterium reproduced every 20 minutes, how many would there be in 2 hours? So here the generation time is already given, which is 20 minutes. And we are asked how many would there be after 2 hours. So we will just first convert 2 hours into minutes. So do cross multiplication, and 2 hours is equivalent to 120 minutes. Now, if the first generation is reproduced in 20 minutes, how many would there be after 120 minutes? So after cross multiplying, x is equals to 6. Since The first generation is 2. 2 to the power of 6 is 64. So there are 64 bacteria in 120 minutes. What if generation time is unknown? We use this formula where G is generation time equals T in minutes or hours divided by N, which is the number of generations. So G equals 120 minutes. That's the time interval in minutes divided by 6, which is the number of generations. So the generation time is 20 minutes. So try solving this by yourself. I hope that you're able to compute on your own. I'm gonna ask this again in our quiz, okay? So what's a bacterial growth curve? When a fresh medium is inoculated with a given number of cells, the population growth is monitored over a period of time, and plotting this data will yield a typical bacterial growth curve. This is what is shown in this graph. This is the typical bacterial growth curve. When bacteria are grown in a test tube, the population of cells almost always exhibits these growth dynamics. First phase, cells initially adjust to the new medium. Lag phase, until they can start dividing regularly by the process of binary fission. That's the logarithmic phase, log phase, or the exponential phase. When their growth becomes limited, the cells stop dividing. That's the third phase, the stationary phase, until eventually they show loss of viability, death phase. So note the parameters on the x and the y-axis. Growth is expressed as a change in the number of viable cells versus time. So let me explain further what happens in each of the phases of the growth cycle. The beginning phase, the lag phase. Immediately after inoculation of the cells into a fresh medium, the population remains temporarily unchanged. Although there is no apparent cell division occurring, the cells may be growing in volume or mass, synthesizing enzymes, proteins, RNA, etc., and increasing in metabolic activity. The length of the lag phase is apparently dependent on a variety of factors, including the size of the inoculum, time necessary to recover from physical damage or shock and transfer, time required for synthesis of essential coenzymes or division factors, and time required for synthesis of new enzymes that are necessary to metabolize the substrates present in the medium. The second phase, exponential phase of growth, is a pattern of balanced growth wherein all the cells are dividing regularly by binary fission. That's why the line or increasing line, and are growing by geometric progression. The cells divide at a constant rate depending upon the composition of the growth medium and the conditions of incubation. What about the third phase, stationary? So exponential growth cannot be continued forever in a closed system or in a test tube or in artificial culture. Population growth is limited by one of three factors: exhaustion of available nutrients. Second would be accumulation of inhibitory metabolites or end products that may be toxic. And third, exhaustion of space, in this case, a lack of biological space. And during this stationary phase, if viable cells are being counted, it cannot be determined whether some cells are dying and an equal number of cells are dividing, or the population of cells has simply stopped growing and dividing. Bacteria that produce secondary metabolites such as antibiotics do so during the stationary phase of the growth cycle. It is during the stationary phase that spore-forming bacteria have to induce or unmask the activity of dozens of genes that may be involved in sporulation process. The death phase. If incubation continues after the population reaches stationary phase, a death phase follows, in which the viable cell population declines. So during the death phase, the number of viable cells decreases geometrically, essentially the reverse of growth during the log phase. Note, if we are counting bacteria by turbidimetric measurements or microscopic counts, the death phase cannot be observed. So just to reiterate, what causes growth to stop or exponential growth to decline and eventually stop? I've already mentioned this. One, there's exhaustion of nutrients, waste product accumulation, since they produce also secondary metabolites, and some of these metabolites are inhibitory, and some end products are actually toxic, and these products can change the pH in the medium. That's why it's no longer optimal for their growth. For any bacterium to be propagated for any purpose, it is necessary to provide the appropriate biochemical and biophysical environment. So when we introduce bacteria into a culture medium, we call it the inoculum. And culture in this context means bacteria are growing. And if there's only a pure or one species or single species growing in that culture medium, it's called a pure culture. And we refer to the macroscopic visible masses of growth arising from a single cell as colony. The biochemical or nutritional environment for bacterial growth is made available in a culture medium, and depending upon the special needs of particular bacteria, a large variety and types of culture media have been developed with different purposes and uses. Culture media are employed in the isolation and maintenance of pure cultures of bacteria, and are also used for identification of bacteria according to their biochemical and physiological properties. The manner in which bacteria are cultivated and the purpose of culture media varies widely. Liquid media these contain no agar, are used for growth of pure batch cultures, while solidified media, they have the component of agar, are used widely for the isolation of pure cultures, for estimating viable bacterial populations, and a variety of other purposes. The usual gelling agent for solid or semi-solid medium is agar. It is a hydrocolloid derived from red algae. And agar is used because of its unique physical properties. It melts at 100 degrees and remains liquid until cooled to 40 degrees Celsius, the temperature at which it gels. And because it cannot be metabolized by most bacteria, hence the medium component, it is relatively inert. It simply holds nutrients that are in aqueous solution. Perfect. Culture media may be classified into several categories depending on their composition or use. A chemically defined or synthetic medium is one in which the exact chemical composition is known, while a non-synthetic medium or complex or undefined medium is one in which the exact chemical constitution of the medium is not known. Defined media are usually composed of pure biochemicals off the shelf. Complex media usually contain complex materials of biological origin such as blood or milk or yeast extract or beef extract, and the exact chemical composition of which is obviously undetermined. A defined medium is a minimal medium if it provides only the exact nutrients, including any growth factors needed by the organism for growth. The use of defined minimal media requires the investigator to know the exact nutritional requirement of the organisms in question. Chemically defined media are of value in studying the minimal nutritional requirements of microorganisms, for enrichment cultures, and for a wide variety of physiological studies, while complex media usually provide the full range of growth factors that may be required by an organism, so they may be more handily used to cultivate unknown bacteria or bacteria whose nutritional requirements are complex. Complex media are usually used for cultivation of bacterial pathogens and other fastidious bacteria. We have many culture media classified according to purpose. A general purpose medium, like the very popular nutrient agar, can grow almost all kinds of bacteria. If bacteria requires blood or any blood components for growth, the media supplemented with such is called an enriched culture medium. Examples are blood agar plate and the chocolate agar plate. So when blood is heated, it turns chocolate brown in color. That's why the name of the agar is chocolate agar plate. If you want to enhance growth of Shigella or Salmonella bacteria in a stool specimen, you may need to inoculate them first to a selenite F broth, or you'd like to enhance growth of bacteria in body fluid specimens, so you use tryptic soy broth. So these broths are called enrichment broths. And most routinely used culture media in the lab are the selective culture media like your eosine methylene blue or EMB and McConkey agar. Why selective? Because only gram-negative bacteria can grow on these media due to the addition of dyes and bile salts, which the gram-positive bacteria are very sensitive. EMB and McConkey can also distinguish between lactose and non-lactose fermenters. So in function, as to function, these are also called differential media. In food microbiology, they do a lot of anaerobic culture. So they use thioglycolate broth or cooked meat broth. And another is when you want to diagnose a gonorrheal disease, specimens are inoculated onto special medium like Thayer-Martin medium. So special medium, this type of special medium, your Thayer-Martin medium or the modified Thayer-Martin medium, can only grow Neisseria species and related species and prevent the growth of other microbes. And to prevent death in bacterial cells during specimen collection, there's a need to place them in a specimen transport culture medium like your Stewart's. A lot of laboratories in the States, and in the UK make use of these transport systems. For antibiotic assays, and the medium actually is made for ease in antibiotic diffusibility. This is the reason why the Miller-Hinton agar is most often used. And for counting or enumeration purposes, your potato dextrose agar or your blood agar plate can be used as well. So a particular culture medium can function both as selective and differential. And I have here two concrete examples. The first one is your eosine and methylene blue. Look at the green metallic sheen of your Escherichia coli in the medium. This will actually differentiate E. coli from the rest of the enteric bacteria. And EMB, like I said before, because of the eosine and methylene blue dyes, plus the addition of bile salts, this will inhibit the growth of micrococci and staphylococci or all other gram-positive bacteria. That's why EMB is both a selective and differential medium. Differential too because if you see colored colonies, pink or red colonies, this means they are lactose fermenters, while the colorless colonies are the non-lactose fermenters. The second example is your Salmonella shigella agar. It will only select these species and prevent the gram-positive as well and other coliform bacteria. Notice the black dot at the center. This means that the organism can produce hydrogen sulfide. Now, in this case, H2S positive gram-negative bacteria, an example of which is your Salmonella typhi. And those that do not produce H2S will not show the black colonies. So since it selects only gram-negative bacteria and prevents the gram-positive, your SSA medium is also both a selective and a differential medium. So a culture medium can also be both enriched and differential, especially if it is blood enriched, like your blood agar plate. It's differential in purpose because it can showcase three types of hemolytic pattern. The non-hemolytic bacteria, the gamma hemolytic, will not exhibit any hemolytic pattern, while the alpha hemolytic bacteria, you would see greening or a greenish discoloration surrounding the colonies. It means there's only partial hemolysis of the red blood cells. While in this picture, you see the complete hemolysis by Staphylococcus aureus. No, it's called the beta hemolytic pattern. So that's why your BAP is both an enriched and differential medium. I've said this before, selenite F is a very good enrichment medium, not to encourage the growth of Salmonella or Shigella species from fecal specimens, and this will inhibit other coliforms and fecal streptococci from growing. In clinical bacteriology, we inoculate specimens onto a culture medium by way of the streaking method. And we streak for the purpose of isolating the organisms. We streak to isolate. So remember that phrase. No? And you will learn more about this when you proceed to methods of identification. My part here is simply to introduce you on what is culture, what what are the different types of culture medium, and more of this, no, in the topic methods of identification. And so my part here is simply to introduce these terms. So we can either use the quaternary 4 times, but what you see here is just the tertiary. You have the first streak, the second streak, and the third streak, wherein in the first streaking, you see mixed growth, an abundance of growth. And then on the second streak, you now see some some individual but still closely positioned growth. But on the third streak, notice now the single colonies. That is what we mean by isolation. So what we strict to isolate. We need these isolated colonies because for sure these isolated colonies belong to a single class of species. So it will be very difficult to identify a bacterium or a species of or the agent of disease if you have a mixed culture. So if in your primary inoculation of your specimen and you see a lot of growth, no? You see yellow colored colonies and red colored colonies, you will have to identify both. So you need to prepare a pure culture of the red colonies and a pure culture of the yellow colonies, and you need to identify. And you can already estimate the growth here. Since the growth reached the tertiary streak, then you can say it's a heavy growth. Heavy growth of, and then if in the process, at the end process, you now can identify after doing biochemical testing and confirmatory testing, then you can just say heavy growth of, and then what species. So that's called the strip plate method. And you will learn more about other inoculation methods in your next lessons. And we can do deep freezing if we want to preserve this culture, especially pure cultures. We can do lyophilization as well, freezing at -54 degrees to -72 degrees, dehydrated in a vacuum to preserve these bacteria for further testing. Now remember, not all pathogenic bacteria can grow in the presence of oxygen. They are actually killed in the presence of atmospheric oxygen. So for anaerobic culture, you need to perform the process, the pre-analytical phase, inside an anaerobic chamber. Or if you don't have an anaerobic chamber, you really need to work very fast and place them in a gas pack system. Sometimes this is the problem. Your doctor would say the infection is really very evident, and yet you're telling me you have no growth. So maybe the pathogen is not meant for aerobic type of culture, and if your laboratory does not perform anaerobic culture method, then you will just have to refer the doctor to a reference laboratory that can perform anaerobic culture methods. So if you don't have the anaerobic chamber, the gas pack system, you see this jar, can grow anaerobe. So there's the palladium catalyst pellets here. This will enhance the action of what's in this packet. This sachet contains sodium bicarbonate and sodium borohydride. This will mix with any oxygen present in this jar so that this will be oxygen free. And there's an indicator if there is still oxygen or not, a color indicator. So this is the gas pack system. Remember too that there are bacteria that are microaerophilic and at the same time capnophilic. They require low oxygen tension but high carbon dioxide concentration in the environment. So you see the candle jar, the candle is lit, it will eventually die off and then emit the carbon dioxide inside the jar. So that's for capnophilic bacteria. So if, for example, your Streptococcus pneumoniae, if you fail to put the blood agar plate inside a candle jar, it will not actually grow. No, you have very poor growth. So remember that too. So uh, in case you don't have the gas pack system because it's kind of bulky if you place it inside your incubator, others use the packet. No? Just a uh, it's just a packet wherein you can uh, place your culture medium, inoculated culture medium, and you see a tube there, you just so once sealed, when you seal the pocket, you just break that glass, and then this will generate the gas required. So that's another method, but more of this, no, in the methods of identification. So we are still talking about bacterial growth, and we refer to the macroscopic, that is visible, growth on the culture medium as colonies. And for a diagnostician or a bacteriologist, you need to be familiar with the colonies, on your medium. You need to know how to describe them, so you can have an idea of what type of microorganisms are present, in the medium. So, say, mucoid, yung mga slimy, slimy, sticky colonies, we call it mucoid colonies. Mucoid because they tend to produce capsules, and the component of capsules, and that's why they look slimy. No? And for smooth colonies, very smooth, just plain smooth, and the rough colonies too. No, but not only the type of colony, this is the consistency. We are describing the colonies based on consistency. So we have three major types: the mucoid, smooth, and rough colonies. But don't forget to describe the color. Very important also to describe the color. When we talk about bacterial growth, we don't really mean growth in terms of size, right? But in terms of number. I mentioned before that you can describe your primarily strict plate, based on the abundance of growth per strict, primary, secondary, tertiary, or quaternary streak. And then you can just describe the growth, whether it's small growth or few growth, or moderate growth, or heavy growth, or moderate to heavy growth. That's a qualitative way of describing the abundance of growth. But if you really want to count it, so we can do direct microscopic counts using so-called counting chambers, but you will have to count it with the use of a microscope. Okay, and we also have the electronic counting chambers. So I have not seen such electronic counting chambers, but we have a simple one, for water bacteriology. And what we do most often in water bacteriology is the uh, counting for colony forming units. no? So more of this in the methods of identification. My part is simply to introduce you to these terms and some of the methods now in diagnostic bacteriology. So that's the end of our lesson 3 part 2. It's all about bacterial growth. So they divide, they multiply by a very simple way, that's binary fission. And once you introduce them to a culture medium, no, Over time, they will present a typical bacterial growth curve. So remember the four phases: the lag, the log, the stationary, and the decline phases. And then I hope you understand, that they divide logarithmically, and the time it takes for them to double their number is called the generation time. Then, of course, how do we grow them? So we need to place them in an appropriate culture, medium, and then we need to know how to describe their growth, you know, in terms of colonies. So thank you. I'll hope to see you again in lesson 4. God bless everyone.